Welcome to EPG Pathshala. Today we will be talking about some of the neurological disorders like epilepsy, Parkinson's and Alzheimer's. The learning outcome of this modules will be that you will be able to learn about the history, incidence, prevalence, etiology, treatment, etc. about these diseases. So, before we understand them, let us just revise that the brain is made up of trillions of cells and it works in a very regulated manner. It has a capacity to change even after it has been made initially in the embryo stage. However, this capacity to change is very prominent in the embryonic stage, but later on slows down. Besides this, there are also regressive developments happening in the brain. Initially, in the embryonic stage, the cell death, the pruning of the synaptic connections help to make new pathways and improve the function of the brain. In the adult brain, it may help in learning, memory, etc. But sometimes these regressive changes like the cell death may become unregulated, and that may lead to pathological conditions or the neurological disorders. And in most of the time, age is one of the important factors which lead to these diseases. So, we will be learning about these diseases and how do they occur in this module. Epilepsy is a neuropsychiatric disorder. It is manifested by seizures. Seizures can originate from a single region or they may be spread to both the hemispheres of the brain. Epileptic activity in the brain can be recorded by EEG. Now that helps in proper diagnosis of the disease. If we look at the historical view, then we see that initially there were lots of myths and superstitions around epilepsy, and it was looked upon as a curse, and according to one of the ancient thoughts, only gods can make someone unconscious. Even today, many of us get quite disturbed by the sight of the epileptic patient. But epilepsy in the current view, we have understood that it is a complex condition caused by a variety of abnormal processes in the brain. And epilepsy is characterized by repeated seizures and it can be detected by EEG. Now, the epileptic EEG is referred to as ictal, and it is marked by spikes. Now, there are different manifestations of epilepsy depending on the part of the brain from where it originates, the age of the individual, the underlying cause of the disease, and how the seizure activity spreads in the brain. And the manifestation of seizure also depends the part of the brain from which it originates, the speed with which it spreads. And it is not necessary that all seizures are epileptic in nature. There can be seizures which are linked to fever, they can be linked to hypoglycemia, they can link to drug abuse, drug withdrawal, metabolic disturbances. Whereas epilepsy, there is a tendency to have recurrent and unprovoked seizures. The diagnosis of epilepsy is that the seizures which are happening, they should be recurrent and unprovoked. So, that differentiates a seizure from any other condition and that which occurs in epilepsy. Now, this epileptic condition could be due to the genetic makeup of a person or could also be encouraged by the environmental factors. And epilepsy is actually a symptom of an underlying brain disorder, as it is multifactorial and there are variety of causes of epilepsy. If we look at the epidemiology, then epilepsy is similar in developing and developed countries. However, epilepsy incidence is more in underprivileged countries rather than in the developed countries. If we look at the prevalence of epilepsy, then it is seen that it is age related. However, it is not the elderly who are suffering from it. High incidence is rather seen in the children within first week of life. But today, since we have better prenatal care and decreased exposure to risk factors, the incidence has declined over the recent years. Classification of epilepsy has been given by an organization, International League Against Epilepsy, and they have classified epilepsy as of focal onset, generalized onset or unknown onset. Amongst that also there are variations, like in the focal onset, there might be a loss of awareness or the person or the patient may be totally aware. There may be motor areas which are affected, in another case the non-motor areas which are affected. Similarly, in the generalized onset also we have two kinds, motor onset and non-motor onset. Besides this, in the motor area also we have tonic, clonic or other motor areas participating. They can be unknown onset, where again there is a motor and non-motor component participating, but they may be unclassified. So, in the focal area, which is also called as the partial seizures, here the changes are happening only at the limited part of the cerebral hemispheres, and in this the patient may or may not lose their consciousness, and it might be possible that the epilepsy attack may spread to the other areas, and they can, again there are sub classification. It could be simple partial, complex partial or partial with secondary generalization. In the simple partial, the consciousness is not hampered. In short, simple partial seizures is also said as SPS. Now, it also depends that this seizure occurs from temporal lobe or it may occur from the parietal lobe. If it occurs from the temporal lobe, there might be fear, hallucination, etc. If it is from the parietal lobe, there might be tingling, somatic illusion, and there could even be sensation of vertigo. In the generalized seizures, the consciousness may be impaired, and this impairment may be the initial manifestations. And here there is involvement of both the hemispheres of the brain. So, motor manifestations are usually bilateral in nature. The ictal EEG, that is the pattern of EEG shown by these patients. Initially they are bilateral and they presumably reflect neuronal discharge which is widespread in both the hemisphere. Generalized seizures may be convulsive or non-convulsive and may vary considerably. The generalized tonic-clonic seizures referred to as GTCS usually occur without warning, and they can be preceded by increasing frequency of another generalized seizure type such as myoclonic jerks. The postictal period characterized by sleepiness and confusion which may last for 20 minutes. Myoclonic seizures are sudden jerks that involve part of the body. Other generalized seizures consist of mainly atonic and tonic seizures. These often consist of sudden loss of body tone or sudden increase in body tone resulting in a fall. These seizures usually occur in more severe epilepsies. So, if we look at the pathophysiology of seizures, basically there is an alteration in the normal balance of inhibition and excitation, which may explain the basic mechanism of focal seizures initiation and propagation. Actually, during a seizure there is a hypersynchronous discharge which may initiate in a single region of cortex and then it will move to the adjacent areas. Seizure initiation is characterized by two events. First, occurrence of high frequency bursts of action potential, and next is hypersynchronization of neuronal population involved in the area where the former event takes place. These summated synchronized bursts from considerable number of neurons produce spike discharge which can be recorded by EEG. These are called ictals. Now, at the level of single neurons, epileptiform activity consists of sustained neuronal depolarization resulting in burst of action potentials and then a rapid repolarization followed by hyperpolarization. This loop sequence is called depolarizing shift due to the influx of extracellular calcium which causes influx of sodium and generation of repetitive action potentials which together result in prolonged depolarization of the neuronal membrane and thus abnormal neuronal firing. In individual neurons, the membrane repolarization fails to cease and nearly continuous high frequency action potentials are generated. The subsequent hyperpolarizing after potential is mediated by GABA receptors and chloride influx. The focal seizures during its development overcome the surrounding inhibition resulting in loss of after hyperpolarization in individual neurons, and thus the seizure begins to spread beyond the origin of focus. So, the current theories which explain how inhibition and excitation can be altered at the network level explain that there could be a disturbed extracellular ion homeostasis which is responsible for the change in energy metabolism. It may change the receptor function or the transmitter uptake. Therefore, epilepsy is a neurological disorder which underlies several abnormal physiological processes that leads to initiation and spreading of seizures. One of the remarkable property of partial seizures is its origin. The abnormal electrical activity originates from the single region. Much of the knowledge about partial seizures has come from the studies of hippocampus. Firstly, as the hippocampus shows remarkable property of plasticity to any stress or injury that it readily undergoes synaptic reorganization and establish connection with other neighboring neuronal population through axonal sprouting. Secondly, the local selective neurodegeneration of neurons in the dentate gyrus decrease the speed forward and speed backward inhibition, which again initiates the abnormal firing or activity in the hippocampus. These mechanisms of hyperexcitability of neuronal network may coexist in the human epileptic brain. Generalized seizures and similar associated seizures are heterogeneous in terms of their underlying etiologies. However, the mechanisms underlying such generalized seizures are not understood. For instance, in childhood absence seizures, an aberrant oscillatory behavior which involves an interaction between GABA receptors and different ion channels within the thalamus may have resulted in generalized seizures. The summated activity of bursts produced by spike when the summated inhibition produces typical spike wave pattern of an absence seizures. If we look at the treatment, we have number of drugs which have been used for anticonvulsions. They may control but may not cure the disease. So, there are number of drugs which have been used for anticonvulsions, and the dosage of these drugs depend on the person's age, overall health and the medical history. And the three major mechanisms which have been recognized through which the treatments have been done is by modulation of voltage gated ion channels, enhancement of GABA mediated inhibitory neurotransmitters, attenuation of glutamate mediated excitatory neurotransmitter. Although medication can control seizures in about 70% of the patients, but unfortunately the drugs have several side effects. Therefore, drug choice depends on many factors, patient's tolerance for the particular drug. Generally, anti-epileptic drugs were initially given at lower doses and then they are subsequently increased gradually for better results. Surgery can also be performed only when seizures fail to cease with the medicine. Another possible method to control seizures is vagus nerve stimulation, where a small generator is implanted under the skin to stimulate the vagus nerve so the intensity of the seizures may decrease. Interestingly, ketogenic diet has also been found to be effective in preventing seizures, especially in children. Seizures are best described as abnormal activity of the brain in terms of its electrical activity. Seizures have been seen to be at a focal point, that is small areas, or they can be widespread pertaining to both the hemisphere. The simple seizures may be because of the removal of inhibition of GABAergic neurons in that particular area, whereas the generalized seizures may be due to the hyperactivity of the thalamocortical areas of the brain. Seizures can be diagnosed by the EEG recordings which show spikes, and they are called as ictals. Seizures should be recurrent and unprovoked to differentiate them from any other kind of seizures. Today, there are several non-invasive tools available to diagnose the different kinds of seizures, and this has led led to development of certain surgical methods by which some of the complex seizures can be taken care of. Parkinson's disease, or in short PD, is one of the most common movement related neurodegenerative disorder. Its common symptoms are akinesia, bradykinesia, increased muscle tone with cognitive impairment and dementia in the advanced stages. Parkinson's disease was described 2 centuries ago in 1817 in the essay on shaking palsy by James Parkinson. Parkinson's disease has become the second most common age-related neurodegenerative disease after the Alzheimer's. The incidence and prevalence of Parkinson increases with age. The average age onset is approximately 60 years. Onset in persons younger than 40 years is not common. But Parkinson hits men more than the women. Parkinson's disease is seen lesser in the developing countries and its prevalence is more in the developed countries, and it is affected by the socio-economic factors and of the population. Parkinson's disease is closely related to basal ganglia. Basal ganglia is a group of nuclei which is located in the deep cerebral hemisphere. It initiates and coordinates voluntary movements. Basal ganglia is also made up of the subset of nuclei, that is caudate and putamen, which together are called as the striatum. They also enclose globus pallidus, which participates in the movement. To understand Parkinson's disease, it is necessary to understand the motor pathway regulation in the brain. It has been seen that there are 2 pathways which regulate the motor movements, the indirect pathway and the direct pathway. The direct pathway starts from the cortex. There is an input on the striatum which is made up of caudate and putamen. The caudate and putamen communicates with globus pallidus interna, and this in turn participates with thalamus, which gives excitatory electrical activity to the cortex. So, that is a direct pathway between cortex, striatum, globulus pallidus interna, thalamus and back to cortex. In the indirect pathway, the cortex communicates with striatum. The striatum on the other hand communicates with globus pallidus externa. The globus pallidus externa participates with subthalamic nucleus. The subthalamic nucleus can interact with globus pallidus interna. So, this is the indirect pathway, and it will again communicate with thalamus, which will go back to cortex. Now, the indirect pathway is the inhibitory pathway. In the Parkinson's disease, there is a loss of dopaminergic neurons in the substantia nigra pars compacta of substantia nigra, which results in diminished dopaminergic input provided by substantia nigra, which causes difficulty in maintaining the transient inhibition from caudate and putamen, and this leads to the movement related disorders in the Parkinson's disease. So, if we look at the relationship of dopamine and Parkinson's disease, then we see that this is the brain disorder which results from the deficiency of a single neurotransmitter, that is the dopamine. In 1950, Arvid Carlson showed that 80% of the brain's dopamine is utilized in the basal ganglia. Dopamine is one of the central major monoamine transmitter in the CNS, which plays several important roles in normal brain activity. It is one of the most important function of dopamine is to coordinate motor activity and to regulate voluntary movement. The majority of the dopaminergic neurons have their cell bodies residing in substantia nigra, while their axons project to striatum, that is the nigrostriatal pathway. The degeneration of these dopaminergic neurons in the substantia nigra pars compacta underlies the Parkinson's disease. Now, it is very difficult to describe the exact mechanism leading to the neurodegeneration in the Parkinson's disease, but there are two important hypotheses which have been put forward as a possible explanation of pathogenesis of Parkinson's disease. According to one hypothesis, which emphasizes on protein misfolding, which is the instrumental to the death of the dopaminergic neuron, while other focuses on mitochondrial dysfunction and subsequent oxidative stress. However, both these factors are not mutually exclusive, as they may interact to cause the pathology and to become the key, and now this has become the key areas of research. The finding that oxidative damage to alpha synuclein can enhance its ability to misfold and aggregate is one example of such interaction. But they are not exclusive in themselves. They may interact and they both may be causing the neurodegeneration, and this has become a point of research. If we look at the misfolding and aggregation of protein, then what we see is that there is an abnormal accumulation of protein in the brain tissue, which may be the key factor which may lead to neurodegeneration, and it may increase the toxicity in the brain. Now, these are so PD is characterized by fibrillar inclusions inside the cell called the Lewy body. These inclusions or protein aggregate cause a sequence of cellular damage, including disruption in protein and axonal trafficking, and sequestering important protein required for cell survival. Now, as we know that cells respond to misfolding by inducing chaperones, but if not properly refolded, the proteins are degraded in the cell. With aging, the cellular function of inducing a variety of chaperone is impaired, as also the activity of the proteasome, which degrades the protein. So, patients with inherited PD have shown to have mutation which causes directly the accumulation of this toxic protein, or indirectly will cause defective protein degradation mechanism that normally removes the misfolded protein. But if not properly refolded, they are targeted for proteasomal degradation by polyubiquitin, and that removes the misfolded proteins. But with aging, the cellular function of inducing chaperones may be impaired. Even the activity of proteasome may be disturbed. This may be due to the mutations present in the patients of PD. Moreover, mitochondrial dysfunction and oxidative stress has also been shown to be increased in the PD patients, as several oxidative stress markers have been shown to be elevated in substantia nigra, and the availability of anti-oxidative machinery is also less in PD brain, and moreover the metabolism of dopamine may also be disturbed. So, dopamine appears to be the pivotal in rendering substantia nigra dopaminergic neurons to be susceptible to oxidative attack. So, if we look at the treatment, they have used drugs to elevate the dopamine levels by using levodopa, which can change into dopamine in the brain. It mimics the actions of dopamine, and there are other drugs also which are given which affect the neurotransmitter levels. Besides this, the treatment of PD includes using anticholinergic drugs because it helps to reduce tremors, muscle stiffness to some extent. This is because the brain may be having more acetylcholine than dopamine. Today, there are also a third category of drugs described by PD, which includes medications that control the non-motor symptoms of the disease such as depression. The other mode of treatment includes removing globus pallidus, removing parts of thalamus. Now, that helps to treat PD, but these procedures are irreversible and it may lead to complications. So, scientists have found some other methods like deep brain stimulations that can mimic the effect of pallidotomy and thalamotomy. With DBS, an electrode is implanted in the brain to prevent the abnormal neuronal firing. The procedure is much safer than the surgery, and electrodes can be removed if the patient does not agree with it. Parkinson's disease, if we summarize, is a complex disease which is related to movement disorders, but it is accompanied with cognitive defects. The symptoms may vary, but some common factors like depression and emotional changes may be common to all these patients. Several therapies have been developed to treat these patients, but lot of research is still going on to understand the etiology of the disease, so that even better methods can be discovered. Today, there are several molecules which are being used to treat the PD patients, but they are still under research, like several nerve growth factors, antioxidants, etc. Thank you.